to the supporting tissue of the nervous system, but take note, we have the central nervous system. So the supporting tissue of the central nervous system is the neuroglia. The neuroglia have two words. We have neuro, which means the nerve cell, and glia, which means glue. So they glue together the nerve cells and the neuroglial cells plus the loose areolar connective tissue. So glia means glue, and the whole of it, we have neuroglia is the supporting tissue of the central nervous system. And these are the non-conducting elements of the nervous tissue. The neuroglia is composed of the following: loose areolar connective tissue, the blood vessels, nervi vasorum, and neuroglial cells. The neuroglia as a whole have 6 functions. So, number 1, structural, mechanical, and metabolic support. So we have here the astrocytes, which are involved here. Then we have the participation in the blood-brain barrier. Again, we have the astrocytes, but there is another neuroglial cell that is also associated in the blood-brain barrier. Then we have the formation of the main sheet of axons. So these are functioned by the oligodendrocytes wrapping around the axon, serving as a main sheet. Then we have isolation of junctional surfaces of synapses. So we have at the end of the axon, we have the terminal button, which is associated with the synapse, wherein it carries or transfer the information from the nerve cell to the receptor cell. Then repair processes following damage or injury to the nerves. So we have the nasal substances involved in cell injury. So we have the process of chromatolysis. And of course, very important, the neuroglia protects the nerve cells wherein it is associated. We have here an illustration of the neuroglia. The black colored cells, these are the neuroglial cells, and the background are the loose areolar connective tissue and the nerve cells. So in here, what is being distinctly presented are the neuroglial cells. And of course, in the loose areolar connective tissue are also the blood vessels that give nourishment to both the nerve cells and the neuroglial cells. In this slide, it presents the relationship between the nerve cell and the surrounding neuroglial cells. So we have here at the center, the orange color, we have the nerve cell, and surrounding the nerve cells are the different neuroglial cells. So we have here the ependymal cells, we have here the astrocytes, which is associated with blood vessels, and we have here the oligodendrocytes forming into a covering of the axon, and the smaller cells are the microglial cells. Let's start first with the first neuroglial cells. We have the ependymal cells. So in the upper right part of the slide, we have the cross-section of the spinal cord, wherein it has a darker area at the center and a lighter area surrounding it. And at the center, there is a small hole which we call the aperture, and this is the central canal. This is continuous from the spinal cord going to the spaces in the brain we call the ventricles. So from the central canal going to the ventricles of the brain, it is lined by ependymal ependymal cells. And along the pathway, we have a pouch-like structure lined with ependymal cells. We have the choroid plexus. So in the lower portion, take note of the shape of the ependymal cells with the cilia. So these are ciliated low columnar to cuboidal cells. So the ependymal cells are low columnar to cuboidal cells derived from embryonic neuroepithelium. So during embryonic stage, the cells are ciliated, but as they mature, some of the cells carry with it to adult life the cilia. And the cilia is very important in facilitating the movement of the cerebrospinal fluid, or the CSF, from the spinal cord going to the brain. The ependymal cells contain abundant mitochondria and bundles of intermediate filaments. In mature brain, the broad bases of the cells appear tapered into long thread-like processes that branch. So we have here an illustration of the ependymal cells. So on the surface, we have the cilia, and at the basal portion, we have the long thread processes that branch. This is the part wherein the ependymal cells are associated with the other neuroglial cells, or associated with the nerve cell. Another illustration of the ependymal cells. So these are the appearance of the ependymal cells when they line the central canal of the spinal cord going to the ventricles of the brain. So you will notice on the surface we have numerous cilia, and on the basal surface we have the slender processes that branch out. We have here an illustration of the spaces within the brain we call the ventricles. So from the central canal going to the brain are spaces which are lined with ependymal cells. So the ventricles of the brain, we have the lateral ventricle, the right lateral, the left lateral ventricle, the third ventricle, the fourth ventricle, and we have the cerebral aqueduct. So all of these are associated with the ependymal cells, and usually you will notice that these spaces are nourished by the CSF passing through the central canal towards the ventricles through the pouch-like structure we call the choroid plexus. So we have here an illustration of the pouch-like structure we call the choroid plexus. So it produces the CSF, and the CSF is the one that gives nourishment to both the spinal cord and the brain cells. So the choroid plexus consists of a small tuft of capillaries surrounded by epithelium, which hangs in the brain ventricles. The capillaries are covered by a layer of simple cuboidal epithelium we call the ependymal cells, which also surrounds the ventricular space. So from the central canal in the spinal cord going to the ventricles of the brain. So we have here an enlarged illustration of the choroid plexus. So focus yourselves at the center of the illustration. It is a blood capillary, wherein the blood capillary is surrounded by ependymal cells. So the blood capillary, the lining of the blood capillary is made up of a single squamous epithelium we call endothelium. So it's a very thin membrane that lines the blood capillaries. So together with the blood plus the CSF that are produced by the choroid plexus, it will circulate from the central canal going to the brain through the ventricles of the brain, giving nourishment both to the cells of the spinal cord and the brain cells. In this illustration, you can see the longitudinal section of the brain. I think this is one of the hemisphere, either right or left hemisphere. So you can see small white arrows that tells us the flow of the nutrients from the central canal going to the spaces in the brain. So we have here in the lower portion of the illustration, we have here the choroid plexus with the CSF. Aside from the CSF, water is added into the circulation as ions and vitamins and other nutrients. So they flow within the spaces of the brain to give nourishment. Let's go into the next type of neuroglial cells. We have the macroglial cells. So there are two cells that are involved here, and collectively they are referred to as macroglia because they are larger than the other neuroglial cells. So the first cell, we have the astrocyte or the star-shaped cells with numerous branching processes. So the astrocytes are of ectodermal origin. They have a large nuclei and darkly stained with granular cytoplasm and considered to be the biggest among the glial cells. We have here an illustration of the astrocyte, the star-shaped neuroglial cells. I would like you to focus at the end of the processes of the astrocyte are the expanded structure which are associated with blood vessels. Another illustration of the astrocyte with its association with the nerve cell and the blood vessel. So again, take note of the expanded structure that is associated to a nerve cell and to a blood vessel. Let's go into the types of astrocyte. The first type is the protoplasmic astrocyte, or sometimes referred to as short-rayed or mossy cells. So the processes of these cells are short and wavy or tortuous, giving off short branches. The cytoplasm within these branches are granular in appearance or granular in nature, and along the processes, the granules produce several spheroidal swellings called gliosomes. Now, the processes of some of the protoplasmic astrocyte may have terminal expansion, which is associated with blood vessels, and these expansions are called the perivascular foot or sucker foot or pedicels. And these expansions are believed to plug the pores of the endothelium of the brain capillaries, serving as blood-brain barrier. Take note of this. The protoplasmic astrocytes are found mainly in the gray matter of the brain and spinal cord and closely associated to nerve cell bodies or to nerve cells. in such sites wherein there is a nerve cell and a protoplasmic astrocyte, the nerve cells are the primary cells, while the protoplasmic astrocytes are the secondary cells serving as satellite cells. The second type of astrocytes, we have the fibrous astrocytes or long-rayed astrocytes or spider cells. So this type of astrocytes has fewer processes that appear longer and more or less straight, with few branches and closely associated with the pia mater and blood vessels, but are separated from this structure by their own basal lamina. So the cytoplasm is euchromatic with few organelles and free ribosomes and glycogen. Embedded within the cytoplasm of these fibrous astrocytes are fibrillar structures. So these are the neurofilaments. Also, there are gliosomes and pedicels in the processes. And take note, the fibrous astrocytes are more numerous in the white matter of both the brain and spinal cord. We have the third type of astrocytes. We have the mixed or the plasmato-fibrous astrocytes. So a mixture of protoplasmic and fibrous astrocytes. So this type of astrocytes are occasionally found at the boundary between gray and white matter. So the protoplasmic astrocyte, their processes entering into the gray matter, they have the protoplasmic character, while those entering the white matter are fibrous in character. That's why they are referred to as mixed astrocytes. Let's go into the function of astrocytes. So the cells are involved in scavenging ions and remnants of neuronal metabolism, such as potassium ions, glutamate, and gamma aminobutyric acid or GABA, accumulated in the environment of the neurons, especially at the node of Ranvier, where they provide cover for the axon. The second important function of astrocytes is they contribute to energy metabolism. Within the cerebral cortex or within the surface of the cerebrum, they release glucose from the stored glycogen when induced by neurotransmitter or induced by epinephrine and vasoactive intestinal peptide or VIP. The third important function of astrocytes is they are involved in the blood-brain barrier. It is because those astrocytes that are located at the periphery of the central nervous system, they form a continuous layer over the blood vessel and assist in maintaining the blood-brain barrier. The second macroglial cells are the oligodendrocytes. So they are of ectodermal origin and they resemble morphologically astrocytes, but they are smaller compared to the astrocytes with dark staining nucleus and a scanty cytoplasm with a very few short processes with few branches. The cytoplasm is dense with a relatively small nucleus, abundant rough endoplasmic reticulum, many free ribosomes and mitochondria, and a conspicuous Golgi complex or Golgi bodies and, microtubules, especially in the perinuclear zone and in the processes of the cell. So in this slide, we have here the illustrations of the different neuroglial cells from ependymal cells to astrocytes and also to oligodendrocytes. So in the lower part, no, of the illustration, to the right part of the illustrations are oligodendrocytes with fewer processes and fewer branching. The oligodendrocytes are classified based on location. So the first one is the interfascicular oligodendrocytes or sheath cells. They are located in row beside bundles of axon, and they are responsible for manufacturing and maintaining the myelin about the axon of the CNS. Take note, axons of the CNS serving to insulate them. A single oligodendrocyte can wrap several axons with segments of myelin. We have here an illustration of an oligodendrocyte wrapping each processes along the axon of nerve cell. Take note, this will take place in the central nervous system. The myelin sheath formation is brought about by the wrapping of the processes of oligodendrocyte around the axon. The second type of oligodendrocytes, we have the satellite oligodendrocytes, and they are subclassified based on what structure they are associated with. We have the perineuronal oligodendrocytes. They actually are associated in the nerve cells in the gray matter of the CNS. While the second one, perivascular satellite, these are oligodendrocytes that are associated with the blood vessels. So they are referred to as perivascular satellite. The third type of neuroglial cells, so again, to recap, we have the ependymal cells, we have the macroglial cells, wherein we have the astrocytes and oligodendrocytes. Then the third one, we have the microglial cells. So they are of mesoderm origin, so they originate from the bone marrow, and they are members of the mononuclear phagocyte system of the body. So the microglial cells, they are scattered throughout the central nervous system or through the spinal cord and the brain. And they are described to be small, dark staining cells that faintly resemble oligodendrocytes with scanty cytoplasm and an oval to triangular nucleus. They are considered to have the most smallest nuclei of all glial cells. So the largest are the astrocytes and the smallest are the microglial cells. The microglial cells have irregular short processes. The cell body and cell processes are covered with spines. Since they are found throughout the central nervous system, and they are part of the phagocytic system of the body, they function as phagocytes in clearing debris and damaged structure in the central nervous system. When activated, the microglial cells, they will act as an antigen-presenting cells, so APC, and secretes cytokines. So they are involved in the immune system of the body. We have here an illustration of the microglial cells with its small size and branching of the processes. Another illustration of the microglial cell in this illustration, so you can see the smallest size of the neuroglial cells. We have the microglial cells. So in comparison with the other neuroglial cells, the microglial cells are really the smallest cell of the neuroglial groups of supporting cells of the nervous system. This illustration shows us the relationship of the microglial cells with the nerve cells. So they are found in between the processes of nerve cells serving as soldiers. So they watch out for any foreign body that will come along in the nerve cells. The fourth neuroglial cell is referred to as the sheath cell of the PNS. So it is not part of the neuroglial cells in the CNS. Sometimes it is referred to also as neurolemocyte or neurilema. So this one cell have flattened nucleus with small Golgi bodies and few mitochondria, and one of its function is to produce myelin. Now, in electron microscopy, it has been revealed that myelin is the plasmalemma of the swan cell organized into a sheath that is wrapped several times around the axon.